Good morning, dear students. This is Dr. Savita Tidame. I welcome you all in the online lecture, and today we'll be studying the chapter number 2, that is Techniques in Cell Biology. Under this chapter, we have completed simple microscope as well as we also studied the compound microscope, and now today we'll study the electron microscope. Right, so electron microscope, let me show you the diagram, rather image of the electron microscope. So this is the instrument of electron microscope. This complete is there, and this is the display unit, means TVs or monitors are attached to this electron microscope, so that the picture, means whatever element which we are observing under this will be magnified on this optical instrument. Now this electron microscope, commonly called as EM, right? Electron microscope, EM, is an optical instrument which utilizes electrons as a source of illumination for observing very minute objects such as with a great magnification. So it is an optical instrument which utilizes the electrons for observation of a very minute object, very minute object, right? So light is not used as a source of illumination. In compound microscope and in a simple microscope, light was used, right? But in this case, instead of a light, what is used? Electrons, right? Short wavelength, very short wavelength electrons are used as a source of illumination, right? And it is a product of a large number of inventions. This electron microscope is product of what? Large number of inventions and discoveries in the physics. It has not come out just within a 1 day or 2 days, but when the scientists in 15th century, they developed one single magnifying lens. After that, they developed the compound microscope, and then this invention was continuously going on, and then this electron microscope come to existence. So this electron microscope can achieve a very high power of resolution, means it can magnify the image with a great magnification, because it uses what? It uses electrons of much shorter wavelength, and these electrons are nothing but with a shorter wavelength that gives a more magnification or clarity to the image. So today there are several types of electron microscopes available in the field of cell biology, or available for the cell biologist. So both the light and electron microscopes are similar in arrangement, and the functions of their components and consist of three major systems, right? It's a simple compound microscope and electron microscope. They are having the similar kind of arrangement. So they are having three main systems, that is illumination system, then the magnification system, and then observation, that is optical system, right? So illumination system, magnification system, and observation or a magnified image system. So illumination system, in the compound microscope, what was the source of illumination? Light was the source of illumination, or tungsten bulb was the source of illumination. In the electron microscope, short wavelength electrons are the source of illumination, right? Okay, after that, let's move ahead and see the composition, or you can say the construction of this electron microscope. But before that, let's see some comparison between the electron and then compound microscope. So in the light microscope or in the compound microscope, visible light is used, right? Visible light is used as a source of illumination. In the electron microscope, what is used as a source of illumination? Electrons. Yes, source of, in case of a light microscope, source of illumination is at the bottom, whereas in case of this electron microscope, source of illumination is at the top, right? Then magnification glass are nothing but, or magnification system comprises of the lenses. In case of a light microscope, magnifying lenses are there, right? So the objective lens is there, optical lens is there, whereas in case of this electron microscope, coils, electromagnetic coils are there, which are used as magnification systems. And then in case of a light microscope, lenses and condensers, objective and ocular lenses are used. Means different types of lenses are used: condenser lens, objective lens, and ocular lens. Instead of that, in case of electron microscope, coils are used. Condenser and objective and projector coils are used, right? So in case of light microscope, image can be seen with the eyes or on the photographic film, but in case of electron microscope, it can be seen on the fluorescent screen or recorded on the photographic film. So in case of a light microscope, what is that? Principle of fixation, staining, embedding, mounting basically are similar, but in case of a electron microscope, the procedure requires a different mechanical, means the procedure is different. Means for the observation of the element, means object, under this compound microscope or light microscope, different procedure is there, and for the observation of those objectives under the electron microscope, different procedure, different protocol is there, right? Now after that, let's see the construction of this electron microscope. So for the construction of electron microscope, electron gun is used, then microscopic column is also there. So electron gun, it is located at the top of the microscope body. Here electron gun is there, which is located at the top of the microscope body, and it is the source of electron, right? Electron gun is used as a source of electron. It consists of a hot tungsten filament. It is a source of electron forming the beam, and the tungsten filament is surrounded by the negatively biased shield with an aperture through which the electron beam is thrown off to the grounded and positive anode, which is located at the bottom. Towards there, the electron beam is being transported, or it has been directed. Then after that, the microscope column is there. Okay, microscope column is there. It consists of an evacuated metal tube, because it is going to pass the electrons, short wavelength electrons. So it is a metal evacuated tube, and it is fixed at the bottom. So that tube is fixed at the bottom and at the top of the electron gun. So the tube is fixed at the bottom as well as at the top. The number of electromagnetic lenses weaving the screen and photographic plate, means this tube is not free. Whatever attachments are there to this tube, it has been going to hold all together, and these parts are aligned one above other. Means a mechanical system, electron gun, or magnifying lenses, everything, means the coils, they are aligned one above the other. And the microscopic column provides a protective shield to the operator from X-rays that are generated when electrons strike the metal surface. So which are those short wavelength electrons? They're going to strike on, because the electrons have the property to radiate in all possible directions, so they strike on all directions. So this metal shield is there, which is going to protect the operator, means a person who is going to operate this microscope, from the harmful X-rays, right? So then electromagnetic lenses or electromagnetic coils are there, right? So these coils corresponded to, let me show you something. Filament, cathode shield is there. This is anode plate, spectrum, objectives. Okay, this is a scanning electron microscope. All right. So electromagnetic lenses or coils, as we said earlier, in case of the electron microscope, electromagnetic lenses are used as a magnifying system, part of a magnifying system. So the coil corresponds to the condenser and objective as well as an ocular lenses of the light microscope, which is built inside it, inside this column. This all means electromagnetic coils are present, and each coil has a coil of the electric wire wound on the hollow metal cylinder. Each coil means that electromagnetic coil is wound around the hollow cylinder and designed in such a way that an electric current passing through the magnetic coil produces an auxiliary symmetrical magnetic field in the center of the lens, and the magnetic field then forces the electron to spiral around a central axis, and this electron beam passes through the microscope column and deflected by a variable degree depending on the current flowing through the coil of the lenses. So in short, it is big complicated, and just simply keep in mind, electron microscope uses electromagnetic coils or electromagnetic lenses, which are nothing but the condenser electromagnetic coil, objective and ocular electromagnetic coils, which serves the function of a magnification system. Then fluorescence screen is there. Say this, for example, fluorescence screen, maybe this and maybe this one. So this fluorescent screen, the electrons are harmful to our eyes, and the magnified image is observed on the fluorescent screen, not directly observed into the microscope, because the electrons, they may enter in our eyes and can cause damage to the retina. So as to avoid this, the fluorescent screen is attached to the electron microscope so that we can view the image on the screen directly. The screen is located with the chemical which by its excitation forms the image as on the television screen. So these screens are used for formation or for the magnification or as a part of a magnification system to observe the magnified image of this electron microscope. Then transformer is also there. This is said to be the transformer. Okay, so the transformer, the electron microscope requires a high voltage current for the electron gun and the electromagnetic coils, right? So for this high voltage transformers, which can boost the voltage from 220 volt to 50 to 100 kilovolt. So transformers are used for directing more amount of electricity or electric current to this one, that is to the electron microscope. Transformers are there. Then vacuum pumps are there, because whenever we're going to run the electron beam, so vacuum must be there. So vacuum pumps are there. Electron microscope requires a vacuum inside the microscope column, right? Inside this microscope column. So this is maintained by diffusion pumps or ion pumps, okay, which can evacuate the column so that the electrons can be run. Then a water cooling system. See, water cooling system is also required. So electron microscope requires a cooling system to prevent overheating of various parts by the pumps and coils, because the pumps and coils requires voltage of a 220 volt electric current is required, and that can cause heating of various parts and coils of this electron microscope. And if it get overheated, then it can destroy the instrument. So as to avoid this, cooling system is there, and the cooling is been done with the help of the water. Nowadays, modern electron microscopes are equipped with the circulating pumps and refrigerators. Okay, so circulating pumps and refrigerators comes inbuilt with them, and the size is reduced because of the electron system. Okay, so this electron microscope is very complex instrument and totally different from that of the compound microscope on its manufacturing basis, I'm saying. Now how it works, right? How this electron microscope works, let's see. So the working of the electron microscope is very much similar to that of the light microscope. The electrons are used for magnification and image formation. So image formation occurs by the electron scattering, and electrons strike the atomic nuclei and get dispersed. What happened? The electron, they strike on the atomic nuclei and get dispersed, and these dispersed electrons forms the image, and the electron image is converted into visible form by projecting on the fluorescent screen, right? So electron is the form of a beam, passes through the condenser coil and then fall on the object. It is like seeing how the light, how through the light microscope, light has been carried through or transferred through the diaphragm and condenser. It falls down on the object, and then the object means the magnified image of the object is been observed by the eye of the observer through the magnification lenses, right? So whatever light beam is falling on the object through the condenser and diaphragm will going to illuminate the object, and the light rays are then further entered into the objective, through which it has been directed to the body tube, and then through the eyepiece the rays will fall down on the eye of the observer, where on the retina the image is formed. So, this is what the compound microscope. Now in the electron microscope, what is that? Electron is in the form of a beam, passes through the condenser coil and then fall on the object. They get scattered, transmitted through the objectives, or transmitted through the object, and passes through the objective coil, which magnifies the image of the object. The projector coil further magnifies the image and project it on the fluorescent screen and or the photographic screen. It's also called as a photographic screen, and this the image formation occurs when the energy of the electron is transformed into visible light through the excitation of the chemical coated of the screen, and those electrons which reaches to the fluorescent screen forms the bright spot, while the area where the electrons do not reach to the screen forms a dark spot. And the electron dispersion is due to the atomic nuclei, which consist of the protons and neutrons, and the higher the atomic number, the greater is the dispersion, right? Higher the atomic number, greater is the dispersion. Biological materials are generally with a low atomic number, so dispersion of the electron is poor, and thus it results into poor contrast in the image formation, right? So on an average it works on the principle of electron scattering. Now in the electron microscope, what would be the magnification? The question comes in everybody's mind. As we said earlier, it comes with a great magnification, but what is that? So objective and projector coils help in magnifying the image formed in the electron microscope. So for getting the maximum magnification, an intermediate coil is fitted between the objective and the projector coil, and this coil further increases the magnification of the object and obtained with the two coils. Okay, if the magnification of the objective coil is 100, projector coil is 200, then we'll get the net magnification of a 20,000, right? And then electron means what? 100 into 200, that becomes a 20,000 times magnification. Okay, so it is simple like that. 10 into 100 becomes, yes, 1000 times, yes, like that. So electron microscope can achieve the magnification of as high as 1,60,000, right? So minimum 20,000 and maximum 1,60,000. So this magnification can be further increased during the microphotography up to, oh my god, it can be increased up to 10 billion times. So but again the image loses its sharpness. So resolving power of the microscope is limited by the wavelength of the illumination and formation of the image. So in short, this electron microscope gives us a very or highly magnified image, right, of the biological material. Now after this, let's see the functions of this electron microscope or applications of the electron microscope. So electron microscope, that is TEM, as different types of electron microscopes are there. So TEM is one of the type of electron microscope, that is a transmission electron microscope, and is ideal for the number of different fields such as life sciences, nanotechnology, medical, biological, material science, forensic analysis, germology, and metallurgy, as well as industry and education. For all these purposes, this transmission electron microscope is been ideal instrument for the study. And further, this transmission electron microscope provides a topographic, morphological, compositional and crystalline information of the object which is under study. So the image allows the researchers to view the sample on the molecular level. So we can get 1,000, not only 1,000, 20,000 times magnification at least. So apart from that, much more, that is 1,60,000, or apart from that again higher magnification can be achieved. So the study of objects, study of molecules, study of the antennae or any other biological specimen at the very molecular level is possible with the help of this microscope, making it possible to analyze the structure and texture of the object under study. This information is useful in the study of the crystals and metals, but also has industrial applications. Then this transmission electron microscope can be used in the semiconductor analysis and production and the manufacturing of the computer and silicon chips. Even the technology companies also used this transmission electron microscope to identify the flaws and fractures, even damages to the micro sizes, objectives. This data can help to fix the problem and also help to make the more durable and efficient product. So for the colleges and universities can utilize this transmission electron microscope for the research studies, and also this electron microscope requires a specialized training, so student can assist their teachers for the operation or for the operating of this transmission electron microscope and also get trained. So students will have the opportunity to observe the nano-sized world in incredible depth as well as in detail, which cannot be possible with the help of a simple compound microscope. But that micro world, or you can say nano world, is quite interesting and easily observed with the help of a transmission electron microscope. After that, SIM, that is a scanning electron microscope. It's again one type of a microscope, electron microscope, scanning electron microscope. So it has again a wide application in different field of sciences as well as in industrial field also. So this SIM, that is a scanning electron microscope, can be used in varieties of industrial, commercial and research applications. So this material science, in case of a material science, SIMs are used in material science for the research, quality control, failure analysis, investigation into the nanotubes, nanofibers, high temperature superconductors are also being studied with the help of this scanning electron microscope. Whether there is any problem related to the quality or the quality check, this microscope can be used. Then in the nanowires for gas sensing. So in the research, researchers are exploiting new ways, that is nanowires can be used as a gas sensors by improving the existing fabrication methods and developing new ones. So this, in that field also, that is in the field of nanowires for the gas sensing, the SIM can be used. Then semiconductor inspection. So reliable performance of the semiconductors requires accurate topographical information, and high resolution three-dimensional image produced by the SIM offers the speedy, accurate measurement of the composition of the semiconductor. So for the preparation of the semiconductor, their all angle study, the SIM will be helpful. And microchip assembly, for the microchip production, this SIM plays very important role, is increasingly relying on this SIM to help gain an insight into the effectiveness of the new production and fabrication methods. Even forensic investigation also involves the use of the scanning electron microscope. The criminal and other forensic investigations utilizes SIM to uncover the evidences and gain further forensic insight. So the uses includes analysis of the gunshot residue, then jewelry examination, bullet making comparison, then print analysis examination, right? So all this can be studied with the help of the SIM microscope or scanning electron microscope. For the biological sciences, in the biological science, SIMs can be used on anything from insect and animal tissues to bacteria and viruses. So insects, all type of insects, their body parts can be clearly and effectively studied with the help of scanning electron microscopes. Even the animal tissues were also studied, as well as bacteria and viruses also studied with the help of this scanning electron microscope. So uses includes the measuring the effect of climate change of the species, right? Climate change of the species, even identifying a new bacterial and virulent strains. As we said, the different strains of a particular viruses are there. How these different strains are there, how can we identify them? Just with the help of the minor variation, minor differences on their morphology as well as genetic component. But morphological differences, how can we analyze? With the help of this SIM, that is scanning electron microscope. We can take the three-dimensional picture of that organism, that virus, and can be studied effectively, and on the basis of that we can segregate them into different strains or different categories. Even vaccination testing, this SIM is also useful. Investigating a new species and work within the field of genetics, this SIM is also helpful. Okay, so electron microscopes, they are very useful instruments and requires huge, means it requires huge money for its purchasing, as well as it has a huge applications, lot of applications are there in every field. We can use it, right? And for the biological sciences, for regular scientific study, as well as for the research purpose up to the genetic level, or morphological differentiation, minor morphological differentiation also been studied with the help of the electron microscope. Okay, so dear students, we have completed the microscopy topic, and in the next lecture we'll be studying the stains principle and composition of stains, as well as different types of stains.